Good afternoon to all. We are highly privileged to have a great and wonderful speaker of international repute, Professor Odeku, as a resource person. Presently, she is rendering her services as Dean, Faculty of Pharmacy, University of Ibadan, Nigeria. She holds a Bachelor of Pharmacy from the Obafemi Awolowo University, LFE, and MSc and PhD degrees in Pharmaceutics from the University of Ibadan, Nigeria. She also has a certificate in the Modern Education, Management and Leadership from the University of Applied Sciences, Osnabrück, Germany. Professor Odeku had her postdoctoral training at the Hebrew University of Jerusalem School of Pharmacy, Jerusalem, Israel. Professor Odeku has been a recipient of many research fellowships and awards, including UNESCO co-sponsored postdoctoral fellowship, MacArthur Foundation grant, CAS TWAS Visiting Scholar Fellowship, TWAS Research Grants, DAD Visiting Fellowship, Alexander von Humboldt Foundation Fellowships, C.V. Raman Senior Fellowship, Research Group Linkage Grant of the Alexander von Humboldt Foundation, and AAU Staff Exchange Award. She is the co-principal investigator, TED Fund National Research Fund, to study genetic and mechanistic basis for memory loss from environmental toxins and old age dementia, the protective role of novel compounds from Nigerian indigenous plants, and holds partnerships for enhanced engagement in research and coach women in science mentoring program, Erasmus program, higher education student and staff mobility interinstitutional agreement. She has been a visiting scientist to several universities. Among them are the University of Manchester School of Pharmacy, Manchester, UK, Shanghai Institute of Materia Medica, Shanghai, China, Martin Luther University, Halle, Germany, and the University of Würzburg, Germany. She also has been a visiting professor to the University of Bonn, Germany, and Kwame Nkrumah University of Science and Technology, Ghana, University of Potsdam, Germany, and Alexandra Stalginskis University, Lithuania, Mazzino University, Kisumu, Kenya, and among others. Her research interest includes drug delivery, nanotechnology, indigenous excipients development, and formulation of herbal medicinal products. She has over 110 articles in books, chapters in books, and peer-reviewed journal articles to her credit. Thank you, ma'am, for accepting our invitation to conduct this webinar. The topic for today's webinar is nanoparticles as delivery system for vaccines. So delegates, I hope this will be a very informative session for you. Enjoy it. And if you have any queries, you may post it in the chat box. And after the session, we may discuss your queries. Now, I welcome Professor Odeku to deliver the lecture. Madam.
Staff and students of the university and other collaborators, both internationally from outside India. It's a good morning here, but I know it's afternoon over there in India because we are 4.5 hours ahead, but it's a delight for me to be here. I just want to stimulate our thoughts and stimulate our interest on this very important topic because it is topical at this particular time in the face of the pandemic when we are looking for a way out and how to get a vaccine for COVID-19. I'm going to talk about that. This is my outline. I hope through the next 50 minutes, I'll be able to stimulate your interest enough to want to look more closely into this all-important subject of vaccine. When you're talking about vaccines, what they do is to stimulate or trigger the body's immune system to produce antibodies against a specific disease. And it could be against bacteria, it could be against virus and any other parasites. I'll talk about that later. And this provides surveillance against future exposure because once you are vaccinated, you can be sure that you are not likely to get that infection again. There are many communicable diseases, the nature of the organism against which the vaccine is being developed. And important properties of any optimal vaccine, because we are saying the vaccine is supposed to stimulate the immune system, okay? But in the process of stimulating the immune system, the vaccine must be safe. In other words, the safety must be such that you are not likely to get exposed to the live organism. We'll talk a bit more about that when we are looking at the different types of vaccines. They must also be very stable because they must not degrade. Having the vaccine must provide that immunity. If the vaccine is not stable and it degrades, then you can know that you can only assume that this patient has been immune or this person has been immune. Whereas if the virus is already, the vaccine is already not active, then the person is exposed to the infection. It must also have the ability to elicit adequate immune response with a minimum number of doses. It mustn't be such that you want to give the vaccine every month. It must be for a period. You want it to be active. And of course, it must also be durable over a prolonged period of time. You don't want the effect to be very short. I know with smallpox, with measles vaccine, the duration is about 6 months, after which the person becomes exposed. And with some other vaccines, you require more than one dose. For instance, we all know the pandemic we have now with SARS-CoV-2. The vaccine, you first have an initial dose, then you have the second dose. So with that, it should be enough to provide you safety and coverage for some period of time. There are different generations of vaccines. The first is the attenuated or killed whole organism. In other words, the microorganism, the basis for the vaccine. And usually with that, then we have the second generation, which are the subunits or the proteins. And then you have the third generation, which are the RNA, DNA, or DNA viruses. But we know that each of these have some disadvantages, because with live attenuated vaccines, it is not, it may be difficult for you to really sufficiently identify the attenuated strains. And of course, with immunocompromised patients, if the immunocompromised patients are more at risk of infection. So if the organism is not really attenuated, it might generate infection. And of course, there's also the risk of the reversion of the attenuated vaccine to a virulent strain. Then you have the inactivated vaccines. In other words, you are using inactivated organism. But with that, you also have the risk of incomplete inactivation of all, which may affect the antigen and of course the immunodeficiency. And then with the subunit vaccines, you have poor immunogenicity, then inability to stimulate cellular responses. And of course, usually they require many booster injections and adjuvants to be able to do it. But with the gene, they are more flexible and they have minimal risk of infection, and but they also face some other challenges, because you have to administer a prime boost, and then in other words, you need a booster dose before the vaccine, so that with this the progress has been continuous. And with different generations, apart from the drawbacks I've listed for each of them, sometimes with the vaccines, in the preparation of the vaccine, you often have low yield. And then the cell cultures are usually costly to maintain. And there's also that danger of the non-virulent strains becoming virulent. And then it is not possible to develop a good culture for all organisms. It is not every organism where you can take a culture for and really culture to use that as to attenuate that and then prepare the vaccine. Example of such is HIV/AIDS. That's why the cure for, the vaccine for AIDS is still out. And of course, with some immunocompromised patients, you really have to be careful. Apart from the challenges with the preparation of the vaccine itself, sometimes it becomes a challenge for you to deliver the vaccine. It's one thing for you to prepare a vaccine. It's another thing entirely for it to be adequately delivered to the sites of action so that they will have maximum effect. So the delivery of vaccine molecules to target sites such as is usually a challenge. And the moment the delivery system, the vaccine will not be stable, it becomes a real challenge because you want good adequate stability. And then of course the requirements with the prime booster dose. In other words, if you can have a vaccine that will release its effect over a long period of time, then that might eliminate the need for a prime boost dose before the vaccine becomes effective. And then there are also, with the formulations, there are challenges with the premature degradation of the molecule and the inability to translate into a functional immunogen. We all know with although I'm using that as an example, with the COVID vaccine now by Pfizer, you have to store at specific temperature, degradation will set in, and of course the vaccine will be a waste. So that requirement for storage is also, to prevent degradation, is also very important. And then the requirement of an adjuvant to potentiate their immunogenicity, for instance, the protein-based vaccine, such as Haemophilus, influenza, and diphtheria, tetanus, and so on, makes it, you need, if you can get a vaccine delivery system that will not require a potentiation, that's adjuvant to do that, then it becomes very important. And it will now be useful. And that is why one of the new generation adjuvants, they classify them all as adjuvants, are the use of nanoparticles as drug delivery system. And the vaccine antigen is either encapsulated as shown here. I hope to see it. This is the nanoparticles. The antigen that you want to vaccinate is either on the surface of the nanoparticles, as you can see here, or they can be encapsulated depending on the nature of the nanoparticulate system and of course the antigen in question and its ability to stimulate the immune system. And so you have these stabilizing agents here to keep the nanoparticles rigid. Okay, and of course they can easily bind to target sites. So you can use the nanoparticulate system to target the antigen to specific sites. And by encapsulating antigenic materials, nanoparticles protect the antigen, which may otherwise degrade rapidly upon injection or induce a short-lived localized immune response. response to degradation. And then the conjugation of the antigen onto nanoparticles also allow presentation of the immunogen to the immune system, similar to the pathogen. You know, the antigen is on the surface, so it's as if the normal organism is entering into specific sites, whereas it is actually the antigen going in, and then you can promote, you can provoke an immune response. And of course, because of their flexibility and their adaptability, you can also deliver them via the non-traditional methods, you know, so that apart from only having them as injectables, you can have oral, you can have inhalations, you can have topical, depending, especially if you are dealing with cancers. Okay, so with nanoparticles, you can formulate the vaccine particles containing vaccine antigen without loss of their biological activity during the formulation process. They are more stable. And then you can also have multiple antigens or targeting ligands and adjuvants. You can have them encapsulated so that you can have one single vaccine protecting from more than one disease. Okay, and also when they are administered orally, the nanoparticles can be designed in a way that it will be protected from the degradation in the intestinal tract. Of course, this will give better and of course the particulate nature also enables the ready delivery of antigenic molecules using surface engineered nanoparticles. So here we have the nanoparticles, that's the green space, and you have the antigen adsorbed onto the surface of the nanoparticle. Okay, when it gets into the antigen presenting cells, the cell will recognize it as normal, similar to its own. So it can be taken in by phagocytosis, so that when it now gets into the cell, it will release the antigen for maximum effect. So you can have targeted delivery, and of course, it will now stimulate the cellular immunity. And you can have the activated cytotoxic lymphocytes, and you have the CD8 and all that, so that you have this stimulation. And because it's sort of, the particles are engineered in such a way that the antigen presenting cells recognizes it as one of its own, the uptake is faster, and of course it's more effective. So you can actually target, so that instead of using, you know, because of the amounts that will be lost during the delivery, most likely what happens is that you have an increase in dose to compensate for the fast biodegradation. But if you can actually target the antigen, you can effectively reduce the dose, and of course, you will increase the efficacy and effectiveness of the vaccine. The advantages of this nanocarrier-based system is that you have improved stability. Then it has very good adjuvant properties. You know, it has that tendency to be able to stabilize the antigen, and it will also assist in targeted delivery of the immunogen to the antigen presenting cell, like I said. And of course, it now protects the antigen from premature protease degradation. And you can manipulate the nanoparticle in such a way that it will elicit sustained delivery, so that the release of the antigen is over a period of time. So it will, instead of having a booster dose, you can actually have one dose, but the release will be slow, such that it will provide that boost that you need for the vaccine to be effective. Then surface absorption can also facilitate their interaction with cognate surface receptors. You know, like I showed earlier, it can interact with the relevant receptors thereby resulting in robust innate humoral, cellular, as well as mucosal immune response when compared to the unconjugated antigen. So that, you know, with the unconjugated antigen, the uptake is usually slower than when you have it as absorbed onto a nanoparticle. And of course, the nanoparticles can also be designed to provoke immune response by either direct immunostimulation of the antigen presenting cells, or they can be used to deliver the antigen. And that is the advantage. You can use materials that could effectively or directly stimulate the immune system, the immune system, as well as deliver the antigen. And for the purpose of this lecture, I'm going to talk about a few of these nano delivery systems, these nanoparticulate delivery systems that have been evaluated for use in vaccine. Now try to go over them. The first are the polymeric nanoparticles. With the polymeric nanoparticles, you are using biodegradable polymers, and they are forming a colloidal, they are colloidal carriers, and the release kinetics is such that when the drug is, the antigen is loaded onto the polymeric nanoparticles, the release rate can be controlled, and you can use a wide range of polymers to do so, including the polyhydroxy acid and the polyamino acids and so on, and other polysaccharides. And that's some of our local materials. They will find usefulness in this regard. And you can use the PLGA and so on and so forth. What you have is, you have a polymeric nanoparticle, you have antigen encapsulated within, okay? And usually you can prepare this by double emulsion solvent evaporation, although there are other methods. And the nature, the release rate can actually be modified by the type of polymer. Sometimes you can have them as oil in water emulsion, so that you can easily prepare by solvent, the normal emulsifying system, and or use of solvent evaporation. And when you use this method is limited, since the antigen efficiency, entrapment efficiency is low. And of course, sometimes when you are using with the oil-water interface, and of course sugars, such as sucrose and trehalose, can also provide stability from denaturation. So, but the most important is for you to have a system that will effectively encapsulate the antigen without the antigen denaturing. So you want it to be stable for that period of time. And of course, you can the main advantage with these polymeric nanoparticles is the ability for you to have a controlled release, so that this system, the antigen is released over a long period of time. The second class of nanocarriers and which are and drug delivery vehicle are the nanoparticles. But we know liposomes, sorry, the liposome. But now we have nanosome, transferosome, biosomes, and so on, depending on the kind of lipids that are used. And you can have the liposomes either as a liposome, essentially, they are self-assembly, but consisting of a phospholipid layer. Okay, you have the phospholipid layer that forms a shell around the aqueous interior. Okay, so that you have your antigen suspended in the aqueous interior, and the phospholipid provides protection. Okay, they can be unilaminar as we have here. They can be multilaminar. In other words, you have several concentric phospholipid shell that is separated by a layer of water, and the antigen is effectively preserved in that interior. And usually biodegradable phospholipids are used. So cholesterol is also used as a stabilizer. It makes the liposomes more rigid. They have been used to deliver vaccine by fusion with target cell membranes, and the structural flexibility and versatility of liposomes are able to encapsulate both hydrophilic and hydrophobic substances. The hydrophilic molecules are incorporated into the aqueous core, while the hydrophobic molecule are incorporated into phospholipid layer, especially you have the multilaminar, the hydrophobic molecule will go into this phospholipid layer, while the hydrophilic molecules will go into the core, hydrophilic. Okay, and studies have shown that delivery of antigen protein entrapped in multilaminar lipid elicits strong T and B beta cell response. Okay, so the antigenic peptides conjugated to phosphatidylserine liposome were readily internalized by the APC cells to potentiate the T helper cell mediated immune response. Okay, because the APC will recognize it, and so the uptake will be faster, and this will potentiate the effect of the antigen and to stimulate immune response, and you will have faster, better responses. And with several liposome-based vaccine nanoformulations have been approved for clinical trials against intracellular pathogens such as some viruses and of course tuberculosis. And one such study already demonstrated the potency of liposomal aerosol carrier in the generation of immune cells against tubercular infections. And this can, and this, and then the lipid DNA complex have been successful to deliver that also in, successfully delivered to the lungs of monkeys. And sorry, I don't know where these arrows are coming from, so disregard it. Then you have the non-degradable nanoparticles. They are non-degradable because the nanoparticles, remember I said with the liposomes, you are using phospholipids that are biodegradable. With the polymeric nanoparticles, you are using polymeric polymers that are biodegradable. But with the non-degradable liposomes, they are you make use of biocompatible non-degradable nanoparticles, nanoparticulate systems such as gold, carbon, and silica. They have been used to generate the shell, so that you have this shell that encapsulates the antigen. It can encapsulate the antigen, can be encapsulated, or the nanoparticles. The antigen they are attached. They provide surface for covalent attachment. This causes, because they are attached on the surface of the non-degradable material, they cause the antigen becomes more stable. There's increase in antigen stability, and of course, they are protected against degradation by proteolytic enzyme. Example of such is the delivery of viral and bacterial antigen using gold nanoparticles, which has been shown to induce a robust host immune response against influenza and immunodeficiency virus. But like I said at the beginning, most of these, some of them are in different stages of clinical trials. I think I'll talk about that. I'll show some of the developments later. This non-degradable nanoparticle encapsulation of plasmid DNA that encode mycobacterium HSP65 antigen on gold nanoparticle has been said to exhibit significant reduction in Mycobacterium tuberculosis. And of course, there are also few studies on the use of hollow mesoporous silica and nanotubes as spherical forms of nanocarbons. All these have been said to improve the immunogenicity and delivery of protein and peptide antigen. You know, protein and peptides are unique because of the fact that they are easily denatured. Any change in the temperature, any change even sometimes in the processing will completely denature them. So you want to be careful when you are preparing such. But the major advantage of these non-degradable inorganic nanoparticles is the low cost of production, and then of course the reproducibility and safety in application. They are found to be safe, so they are very good potential to be explored in the delivery of vaccines. The next are the colloidal saponin containing micelle that is known as ISCOMs. They have been used as self-adjuvanting vaccine delivery system. Two types of ISCOM has been described, both of which consist of cholesterol phospholipid, typically either phosphatidylethanolamin or phosphatidycholine, and saponin. Most often it's the choline saponin. And what they do, the saponin acts as the emulsifying agent, and you say they are self-assembly at ratio of 1 to 5 when you look at the ratio of cholesterol phospholipid to saponin, or for matrices, and of course 1 to 1 to 5 for classical ISCOM, forming in the presence of non-ionic detergent. So look at it. Here is you have the monomer in solution, okay? And you have, the saponin they act as to reduce the interfacial tension, okay? And of course, they act as like an emulsifying agent. And so that you have this aggregate of micelle, okay? And depending on the nature of the antigen, the antigen can be entrapped either within or on, okay? So because it's the non-ionic detergent, which is then removed. Once you have the micelles formed, you can remove by dialysis or ultracentrifugation. So this results in a complex pentagonal dodecahedron arrangement of micelles containing saponins and lipids held together by hydrophobic interaction and stabilized through its negative surface charge. And that negative surface charge offers the advantage of the antigen can adhere to the surface. It is used to entrap viral envelope proteins such as herpes simplex virus type 1, hepatitis B, and influenza. And the protein from the range of bacteria and parasites, including E. coli, Brucellosis aeruginosa, and so on, have also been used to self-assembly ISCOM. And of course, they have the advantage of that stability. The next class are the dendrimers. The dendrimers are three-dimensional, monodispersed, hyperbranched nanostructure that are made up of a mixture of amines and amines. And this is a figure of work, okay? So you have the core here, which you can use to encapsulate small molecules, or they could be polymers, they could be dendrimers, or so on. And then you have a network. You can see the complex network, and they are arranged into different generations. So you can have one here, you can have another, so that these void spaces in between makes room, is the room you need to load your molecule, your antigen, okay? And the interior branches, that is the branches here, is robust and provides a covalent structure to connect the core to the surface group. And the surface groups also carry a surface charge. They could be cationic, anionic, or hydrophobic. They are biocompatible, and they can be used to target direct groups. So they have that advantage of that stability, okay, because of the complex nature. But a few studies have explored the application of dendrimers in the delivery of different antigenic molecules. The most commonly used dendrimer for vaccine are the polypropyl imine and the polyamidoimine. They have been used as dendrimers. A single dose of the dendrimer encapsulated multiple antigen was found to produce strong antibody and T-cell response against Ebola virus and influenza and Toxoplasma gondii. So this generation of robust immune response was found to be due to the efficient uptake of the dendrimer by the host cell. You know, the nature allows that, you know, the nature of the polymers and the fact that, of the dendrimers, and the fact that you can actually load the dendrimers with the molecule, and of course the whole cell will recognize it, so the uptake is higher. So that also will improve the activity. There are also other studies that have been done on the vaccine efficacy of HIV transactivator of transcription. It's based on the DNA and the use of dendrimers. And of course, the dendrimers can also be tailor-made to attain certain biological and physiochemical properties. They can be used to conjugate several ligands to single molecules. In other words, you can have different antigens, and they are said to be a promising candidate for the development of new generation vaccines with enhanced immunogenic properties. So all these, there are several studies that are going on this. Then we have the virus-like particles. And the virus-like particles, you can see they actually looks like the virus, okay? It has the capsid and then the glycoprotein, and the antigenic glycoprotein can adhere to it, and then the viral lipid membrane is here. These virus-like particles are molecules that closely resemble viruses, but they are non-infectious because they contain non-viral genetic material, and they can be made of naturally occurring or synthesized. They can be naturally occurring or synthesized through self-assembly of virus-like structures. And they are various, they are very similar to those. Typically, they're the stage range of 20 to 150 nanometers. Viral lipid particles have been said to trigger immune system in the body without the symptoms of virus, okay? And they are manufactured in such a way that they provide they can be made from bacterial, yeast, insects, or mammalian cells, okay? And their structure is such that they have the structural proteins is similar, and of course you can have the antigenic protein, you can have the natural lipids, you can have the non-infective generic molecules, depending on the kind of organism you want, the kind of antigen you want to encapsulate, okay? And they are attracting interest because of their ease of production and the ability to stimulate immune response. And unlike viruses, the virus-like lipid assemble without encapsulating any viral RNA, meaning they are non-replicating and non-infectious, okay? And what is done with this is sometimes a mixture of lipids that closely mimic the properties of the virus is used for this. But as we know, this is almost the same system that is used with the attenuated vaccines. The implication of this in production, in the development of vaccine, is that with nanoparticles, you can have targeted delivery of nanoparticles, which can activate innate and adaptive immune response. You know, so the nanoparticles can function in stimulating that the innate ability. And you know, because of their unique properties, you know, usually the sizes are nano size, they are small, then they can be made to carry surface charge, and then their hydrophobicity and the surface topography, and of course the material composition can also be optimized to ensure that the nanoparticles to ensure improved optic of the nanoparticles, okay? And of course, the nanoparticles can also penetrate extracellular matrices so that they can travel to lymph node, to where they can be internalized by resident macrophages to activate the T cell responses. So this has made them very useful in nanoparticulate delivery system. They can also be used to increase cross-antigen presentation, and they can also be used as adjuvants to generate immune response in lymphoid organs, such as the lymph nodes, like I said earlier. There are some nanocarriers and nanocarrier-based vaccines that have been developed. Some are being tested, some are still on the at the experimental stage, okay? And we'll just go through a few. You have some of them for anthrax. You have the use of antigenic proteins, and what is used there is the polylactic glycoacid nanospheres. Then you have the chitosan nanoparticles. The chitosan nanoparticles belongs to the polymeric class, the polymeric nanoparticles, because it uses the chitosan, which is a polymer, and it uses the DNA encoding T-cell epitope, which is incorporated into it, and it's being tested against tuberculosis. Then there are also the mycobacterium lipids that have been incorporated into chitosan nanoparticles for tuberculosis. Then there are various polysaccharides that have been used as liposomes for pneumonia and cholera, Helicobacter pylori, cystic fibrosis, anthrax, and so on. So you can see there are quite a number of bacterial infections where that are being evaluated, because if you can have a formulation where instead of using the attenuated organism, especially when it is difficult for you to culture, and when some viral infections that have also been used, such as hepatitis, novel virus infection, Newcastle disease, especially with animals, human papillomavirus, and so on. So there are various formulations that are actually, some of them are being developed for these viral infections. And okay, so I want to talk very briefly just to stimulate our interest, because it's I think it should be the topic for another webinar. The case of SARS-CoV-2 vaccine. We know the severe acute respiratory syndrome coronavirus has been with us now for over a year. It's almost a year ago when most countries, when Nigeria went into lockdown, and I'm sure in other countries it's even longer. It started in Wuhan in late 2019, and has spread very widely globally, with over 110 million cases and 2.5 million deaths. And it's still increasing, but thank God the vaccines have been, some vaccines have been developed and they are being used currently. So the vaccine candidates against SARS-CoV-2 are mainly based on the viral spike protein due to its vital role in viral infectivity. And there are over 200 potential vaccines at various stages of development. At least 48 of those vaccines have been tested in experiments with human volunteers. And this is the structure of the SARS-CoV-2 virus. Okay, so you have the spike. So looking at the structure, what the attempt that is being done is to look at what will replicate this structure, one. Two, what is going to improve the binding of the vaccine antigen to the virus to destroy it and stimulate that immune response, so that the host cell can be protected. And there are various technologies. I've got them out. There's the genetic coded vaccine, the Pfizer and biotech, and then the Moderna is based on the RNA DNA strand. And then you have the one from AstraZeneca, is the vector vaccine, where you have the viral vector, and this DNA is not is inactivated, so it recognizes it as to stimulate it recognizes it as if the virus is injected, so as to stimulate the immune response. Then there are some, the Novavax vaccine consists of protein viral fragments, so it makes use of the protein, the viral protein. Okay, and then the one by Sinopharm is the weakened or inactivated vaccine. So you can see different companies are looking at different, it is still novel. Like it has been said, so many things are still unknown about this virus. And because of that, so many things are still unknown. The potential of even these vaccines to stimulate the immune system. For instance, one of the questions people have been asking is that indeed you are vaccinated, you receive the first dose, you receive the second dose. For how long will you be provided immunity? Is it able to stimulate for your immunity? Is it for a lifetime, for 6 months, for 2 months? So many things are still unknown, but because of the emergency nature, so many studies have been going on. I don't know whether you have this controversy in Nigeria. Sorry, like we have in Nigeria, in India. People have been saying, oh, this virus is encoded because it is RNA, DNA, that is the RNA that is encoded into the vaccine, that you have nanoparticles, nanobots that can actually monitor you, that they want to use it to monitor you. I don't know whether you have that controversy in India, that so they are trying to track and reduce the population. And so initially at the beginning, there were a lot of campaign about why people should not receive the vaccine against coronavirus. But I think it's an ongoing debate that it's only when it is proven that that is probably when we'll be able to know so many other things about it. So in conclusion, nanotechnology-based vaccine delivery system have been established as an efficient antigen delivery system to augment immunity against various diseases due to their small size, their large surface area to mass ratio, their stability, aptitude for surface modification, and their control rate of degradation to allow for sustained release of antigen. And nanotechnology-based vaccine delivery system will be highly dependent on the development of safe, non-toxic, and non-immunogenic nanocarriers, because whatever you're using in the host when it is injected, and the wide variety of nanoparticles that have been described, some of which are highlighted, but there are still a lot of safety concern, because some people also have raised the fact that because you are using nano-sized particles, the tendency is that the particles, the absorption will be higher. So you will have very high concentration. So the question is, will that the accumulation and of course the high concentration, will it be safe at the to the patient? But like I said, toxicity concern, of course, and the fact that they can accumulate in the cell, because there's better uptake. Is there going to be accumulation of these nanoparticles in the cells for a long period of time? So there are still so many unanswered questions, but it's good to know that they form a potentially good and efficient source for the delivery of vaccines. And these are some of the references. I want to thank you for your kind attention. Thank you very much.
Thank you so much, ma'am. The session is been open for question and answers. So let's go with the questions. So, ma'am, some of our delegates has asked some questions. So
I hope my presentation was not too fast.
No, ma'am, it was very good. It was very good. Hope we have more presentation from you in the future.
Thank you.
So some of our delegates has some questions. So first one, it's the questions, how safe are the nanoparticles in modern vaccine?
Okay, I think it's Moderna.
Moderna's vaccine.
By Moderna, yeah.
Yeah.
Like I said, there's been, so many things are not known, like I said at the beginning. And studies have shown, it's like with similar to so many other drugs. Usually, you measure the safety, the benefits stroke safety ratio. Sorry, the benefits stroke toxicity ratio. You want to see how many people are likely to benefit. But you know, with some people, you can have anaphylactic reaction that could be limited to very few individuals. Okay, so
Okay.
even though the company has brought out the safety program and say, oh, it is safe, but in how many percent of the population? Is it in 99%? There are so I think, but from the data prevent it from having some adverse effect in some patients, but you want to look at the risk benefits. Okay, thank you.
Okay. So the next question is, can nanoparticle filled with drugs instead of antigen can go to specific site with cancer cell and just destroy the cancer cell?
Pardon?
Can nanoparticle filled with drugs instead of antigen can go to the specific site with cancer cell and destroy the cancer cell? Means the cancer.
Okay, with cancer, yes. With cancer,
yeah.
There are so many studies that have used nanoparticles for cancer. You know, cancer is one of the most deadly diseases now that we have. So it's one of the most researched area with nanoparticles for cancer cells. And there has been successes because, you know, because of the toxicity of drugs that are used for cancer. Nanoparticles will offer, because, you know, I was talking about the fact that with some of them, they can carry a surface charge. And this surface charge can be used with the cancer cells so that it attaches itself to the surface of the cancer and, of course, destroy it. With that, you are able to have lower dose, and of course, you are most likely to have better efficacy.
Okay. So, ma'am, the next question: whether aluminum nanoparticles vaccine adjuvant delivery system be able to safely elicit systemic immunity?
To elicit, yes, there have been studies to show that yes, they do. But you know, with usually with such formulations, you will need the fact that it successfully does so in small group of patients, of volunteers. You have to increase the number of people that you are administering it to during the test. So this will also improve the efficacy, but the most important is to try to find that balance where you have the efficacy and safety.
Okay, ma'am. So we have a few more questions. Why was the RNA or DNA vaccine preferred for use in the COVID case over the traditional vaccine?
The RNA DNA vaccines are an improvement on the traditional vaccine, vaccines, okay? Because what you are taking is the gene, but this time it cannot cause disease. It is an inactivated. So it is said to be, it can be more effective. And then, you know, because so many things are known about the virus itself, the fact that if you want to use the traditional method, the attenuated vaccine, in other words, you are taking the virus, you are attenuating it, and then injecting it. What will happen is it going to be effective over a long period of time? You remember when I was talking about it, I talked about the fact that sometimes it can be reversed and it can become active and can produce disease. And I think this is even important because with SARS-CoV-2, we now have new strains developing. In other words, it is developing and different mutations. It is mutating at a very fast rate. So it's, I think among all those that were tested, the mRNA DNA appears to be most effective. Even with those that have done vaccination, that have been vaccinated, they feel that is has been very more effective. But as you know, I'm sure you know that most of these companies will have tried different methods before they come out with the safest and the most effective.
So,
I hope I've answered that question.
Susanna, question. Is it the first time lipid nanoparticle based delivery system been used?
Is it the first time? No. There are so many lipid nanoparticles that have been used. It's not the first time.
Okay.
And in fact, liposomes are one of the very few, very first sets of nanoparticles that have been developed, that were developed.
Okay, so there's another question. So regarding vaccination, what's the status in Nigeria?
Presently, we don't have vaccine. Okay. There's no vaccine in Nigeria. The government is in the process of importing the vaccine. And I think they have been talking about the fact that they are importing from India. So, but we are not sure yet. But we don't have vaccine. Okay. Those that have been vaccinated in Nigeria probably got their own shots in Europe or from UAE, United Arab Emirates.
Okay. And so here's another question. Are nanoparticle-based delivery always targeted?
No, they are not always targeted. Okay. But you can, they have that, you can modulate it. Depending on what you want to achieve, you can actually modulate it. And but they offer that advantage because, you know, with the delivery system, you have to look at what do you want to improve. What advantage can the nanoparticles particulate system offer? Okay, and before that of course will determine what kind of nanoparticulate systems you will use, and of course the kind of disease you are targeting. So you are going to consider quite a number of things before you finally settle for that.
Ma'am, and the question, can nanoparticles included in dry powder inhalations for pulmonary vaccine?
Yes, in dry powder inhalation, yes, they can, because what they offer is the advantage of they will not be easily degradable, and of course the drugs, the antigen will be stable.
Okay.
So, and because they are particulate systems already, you can modulate the size to determine what size range, especially for inhalation, what size range will easily go into, if we are targeting into the lung, what size range will can easily be inhaled to get to the target site. So you can easily modulate that.
Okay. So, okay, so with that, we close the Q&A session. Now, okay. Finally, thank you, ma'am. Okay. So, ma'am, that was a very impressive session. Thank you for your valuable time and sharing your knowledge to all the participants over here. Thank you so much. We are looking forward to another session, maybe in future. Thank you. Thank you a lot. And also thank you for all those who had participated in this webinar. Once again, thank you and have a nice day.
Okay, ma'am. Thank you.
Yeah. Thank you very much. Thank you for having me.
It's a pleasure. Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am. Thank you.